Alright. Alright, hello. Today we're going to be talking about the drug Xigris, or known by its trade name, Xigris. I'm Philip Chapman, and this is my partner, Spencer.
So,
Xigris is a brand name for Xigris FL. It's a recombinant form of the human activated protein C. The drug Xigris is made of ratio of activated protein C, salt, and a bulking agent. The drug is said to be lyophilized in the powder form and is reconstructed using sterile water or sterile saline. It's intended to treat sepsis.
what is activated protein C? Activated protein C is a naturally occurring anticoagulant. It regulates uh, this homeostasis by inactivating factors 5A and 8A through the steps of the coagulation cascade. It is made in vivo inside the liver through a single polypeptide.
The manufacturer of Xigris is very complex since activated protein C acts as an anticoagulant and an anti-fibrotic agent. Because of the activated protein C can other degrade, decreasing its effectiveness as anticoagulant. The manufacturer aims to reduce the activated protein C autoregulation through the use of bulking agents.
Bulking agents. In order to achieve the activated protein C autodegradation, manufacturing uses bulking agents. There were four bulking agents used that consisted of mannitol, trehalose, raffinose, and sucrose. The addition of bulking agents allows the drug to reach a stable formulation with a unique appearance that can also be readily solubilized with an appropriate solute, such as sterilized water or saline. The formulation is said to be 1 part of activated protein C and 1 to 10 parts of the bulking agent based on the weight to weight basis. The bulking agent is an important variable in the formulation or the freeze drying process. The presence of the bulking agent with the activated protein C is said to provide chemical and physical stability.
Buffer. In order to be in the desired pH range, the activated protein C contains a buffer solution for staging in the pH range. The buffer systems can include trace acetate, sodium citrate, or sodium phosphate. The molarity of the buffer should be around 10 millimolar to 50 millimolar.
Ionic strength. Ionic strength is important in order to ensure the solution's stability. The ionic strength is said to be determined by the salt concentration of the solution. Two salts typically used are potassium chloride and sodium chloride. During freeze drying, the salt concentration must be high enough to ensure that it will crystallize.
Preclinical trials on baboons. The baboon study showed that the activated protein C can function as an effective in vivo anticoagulant without causing severe bleeding complications. In addition to preventing cholestatic response, which follows infusion of E. coli, activated protein C also prevents lethal effects.
Preclinical trials on dogs. In the study that included 22 dogs, the dogs were selected to receive one of three doses of activated protein C that would be administered intravenously for 2 hours. The dogs went through a procedure to include a thrombus within the left circumflex coronary artery. The clotting, bleeding, activated partial thromboplastin time, APPT, and prothrombin times were all measured before the administration of the drug, 60 and 120 minutes into the administration of the drug. The quote, dose-dependent increase in APTT was observed. Significantly elevated bleeding times were noted only in the 1.0 milligram per kilogram hour group, with peak increases seen 60 minutes into the drug infusion, end quote. The COI 2004, the long-standing effects on bleeding time were not observed since the animals sacrificed 120 minutes after the infusion of the drug.
The study included 22 dogs that were selected to receive one of three doses. Clinical trials. The drug used in double-blind phase 3 clinical trial had 1,690 patients with severe sepsis. In clinical trials, the data showed that during trials, what the patients received was either the drug sulfin alpha activated or the placebo with 1:1 manner ratio. The trials also used block randomization, and all drug assignments were made through Central Randomization Center. The dose was called for 24 milligram per kilogram body weight per hour, and it was administered through intravenous line at a constant rate in foil wrap bags for 96 hours. This is the activated protein C, how it works, and what happens when it goes inside. And as you can see, once the activated protein C goes inside, it goes through inhibition through the tissue factor, and then goes to collagen cascade, and then goes into the factors as well. And the factors, it like turns into a bunch of things such as proopulin, which suspends fibrinolysis, which leads to fibrinogen clot. Or the other way it can go is through insulin 6 or insulin 1 factors, which reduces the reduction of rolling, leading also to the same factor, fibrinogen clot.
Clinical trials continued. Patients were accessed blood samples that were obtained for 4 times and were assessed for D-dimer levels and interleukin 6 levels. Blood samples that measured neutralizing antibodies against activated protein C were collected on days 14 and 28 or during the discharge from the hospital.
Clinical trial result. The efficacy of the drug results found 28 days after the infusion of the drug, or placebo, was that in the placebo group, 259 of the 840 patients, or about 30.8% had died. In the drug group, 210 of the 850 patients, or about 24.7%, had died. This showed that the rate of death between the two groups was significant and reduced the risk of death by 6.1%. And the amount of patients that had the adverse effect was higher in the drug group than in the placebo group by 3.5% for the drug group and 2.0% for the placebo group. The patients that received the drug and those who received the placebo had similar bleeding events. Both groups had an identifiable predisposition to bleeding. 2 patients who had received a drug had a fatal intracranial hemorrhage during the infusion. Additionally, 1 patient in the placebo group also had a fatal intracranial hemorrhage. Between the 2 groups, the rate of pneumocophic events was similar. The new infections occurred at 25.5% for patients who received the drug, and in the placebo group, it occurred at a rate of 25.1% for those who received the placebo.
What is sepsis? According to CDC, sepsis is the body's extreme response to an infection. It is a life-threatening medical emergency. This means that when someone gets an infection, it is usually due to an infection, one of many places in the human body, such as the skin. The CDC says that without timely treatment, sepsis can rapidly lead to tissue damage, organ failure, and death.
How is sepsis diagnosed? Sepsis is usually diagnosed using the secondary organ failure assessment, or known as SOFA, and a quick SOFA is the diagnosis stage determined by having number one, respiratory rate less than or equal to 22 per minute. Glasgow Coma Scale greater than 15. And systolic blood pressure less than or equal to 100 millimeter per Hg. The patients who meet two or more of the quick SOFA criteria are to be investigated for report organ dysfunction and consider escalation of therapy and evaluate for referral to critical care.
Sepsis diagnosis continued. The patient is said to be undergoing a septic shock when there is a need for vasopressor therapy to maintain a mean arterial pressure less than or equal to 65 millimeters. What is that exactly?
Hg.
Is that yeah? But what is HG an acronym for?
Mm-hmm. I think so.
Okay, and then a serum lactate. Uh, uh, less than two millimoles, persisting after adequate fluid resuscitation.
The causes of sepsis. Therefore, pathologic pathways to determine sepsis. Number one, ileal dysfunction. The ileal activation increases the expression on the number of lethal lesions. With the increased lethal transmission into the tissues and the permeability of the epithelium is also increased, in the lung leading to intestinal biliary embella and in the gut increasing bacterial translocation, potentially exacerbating the inflammatory cascade already initiated by microbial products. Calligraphy. Altered coagulation is extremely common in sepsis. Immunological damage removes the protective function of the natural anticoagulant protein C pathway and converts the epithelium into a proformic surface. In addition, bacterial products and inflammatory cytokines activate tissue factor, the main inhibitor of the extrusive pathway of blood coagulation. This proformic state may lead to blockage of the macrovectors as well as giving the rise to consumption collapsively discriminated in vascular collagen. Gram-positive products can also directly elevate the contact clotting system. Causes of sepsis continue. Cellular dysfunction. The most severe cases of lethal sepsis. Autopsies studies show that little evidence of cell death despite widespread organ dysfunction. The molecular basis of this is still not clear, although generalized reaction in energy species by itself suggests some kind of hibernation-like process. Codominant within this elation in cellular function and numerous metabolic changes, notably increased catabolism, insulin resistance, and hyperglycemia. Cardiovascular dysfunction. Many studies show that patients with sepsis have a decreased systemic vascular resistance (SVR) with a normal or increased cardiac output. The so-called hyperdemic state of sepsis cardiac output is maintained at the expense of left ventricular dilation with a reduced ejection fraction and a reduced left ventricular stroke work index in response to left bin color, and diagnostic volume increase. These changes can lead to hypertension characterized as septic shock.
Pathways to sepsis and multi-organ failure. The key pathophysiological changes of sepsis and how these combine to produce multi-organ failure. So here's a diagram that shows sepsis leads to endothelial dysfunction, coagulopathy, cellular dysfunction, cardiovascular dysfunction, leading to capillary leakage, disseminated intravascular coagulation, reduced tissue perfusion, catabolic state, reduced cellular energy consumption, left ventricular dilation, hypotension, and then finally multi-organ failure.
How is Xigris used to treat sepsis? The treatment of sepsis using Xigris goes as following. A vial of 5 milligrams or 20 milligrams is selected. A dose of 24 milligrams per kilogram per hour is calculated for body weight in kilograms. The drug is then reconstituted using sterile water. The infusion is given to a patient using intravenous line for 96 hours. Conditions include hypersensitivity to atostasis and any excesses or dominant from a manufacturing process. The influences that included are sucrose, sodium chloride, sodium citrate, citric acid, hydrochloric acid, and sodium hydroxide.
Marketing. Eli Lilly contracted Belzito and Company 2002 to aid in their marketing campaign for Xigris. Belzito and Co. would implement marketing strategies many would later criticize as aggressive. Eli Lilly contributed 1.8 million to the creation of the values, ethics, and rationing and critical care VERIC task force. Although VERIC states that its results are independent of major bodies, received unrestricted funding from Eli Lilly, which raises questions about its ethical integrity, potentially allowing commercial interest to override objective empirical data in care and treatment strategies addressing potentially life-threatening fatal illnesses. VERIC seemed to have a disproportionate effect on the creation of subsequent guidelines, which proposed activated human protein C as a viable treatment path for ICU patients dealing with sepsis. Eli Lilly had contributed substantially to the Surviving Sepsis campaign, which came with the added benefit of the recommendations by physicians and the acceptance of Xigris into the mainstream clinical settings.
Lawsuits. Before Xigris suffered NFL, it became available for patients and applicable clinical use. A lawsuit was filed on behalf of Alfred Pharmaceuticals against Eli Lilly, alleging a valid infringement. In 2002, Ariad, along with Massachusetts Institute of Technology, the Whitehead Institute for Biomedical Research, and Harvard College, filed a lawsuit against Eli Lilly. They alleged that Lilly sells Xigris stuff in the flower and evades the rafloxacin, infringe on U.S. patent suits 410, 516, the 516 patent, transition factor NFKB, regulates gene expression, which is subject to 516 patent. The 516 patent creators were first to recognize SFKB and discover how it regulates gene expression and its relationship to the body regarding the immune response, triggered by presence of intruders in the body, such as lipokine from bacterial agent, is released from its inhibitor. The SFKB then enters the cell nucleus, where it attaches itself to genes that have NFKB recognition site and triggers their transcription. The genes that spawn from this AT cyclides aid the body in eradicating the extracellular intruders. However, the response is too great, the results could be catastrophic. Seizing this opportunity to possibly cure life-threatening illness, several ballots were filed regarding NFKB. Thus, Eli Lilly was found liable because both Xigris and Evista were trying to achieve the substantial admission of NFKB. Lilly, however, contested this decision and sought to overturn a U.S. Court of Appeals for the Federal Circuit. In 2010, Alfred's claim was overturned and his path was very invalidated.
Reason that Xigris was withdrawn was due to its lack of efficacy, according to the European Medicine Agency's Committee for Medicinal Products for Human Use, CHMP, in 2007. The CHMP found that the initial efficacy results of the pivotal clinical study, the PROWL study, could not be reproduced. In order for CHIMP to conduct a benefit risk balance of Xigris, CHIMP requested Eli Lilly to conduct a new placebo controlled clinical study called PROWL Shock to confirm that the benefits of Xigris outweighed its risk in patients with septic shock. The results of the PROWL Shock study failed to meet the endpoint reduction for 28-day all-cause mortality in patients treated with Xigris and those treated with placebo. Because of the results of the study, the drug was withdrawn.
All right, this is our references for the drug. Feel free to look at it. All right, thank you for listening to our presentation. We hope you enjoyed our presentation about Xigris, and you have a great day.